Hi everyone, my name is Mr. Anderson, and I am mean to people on the internet. I'm always very polite about it, though. I just tend to ask some pretty tough questions. So if that's something that you guys like watching, or something you want to learn how to do yourself, then you might enjoy my content. This is probably going to be the first piece of long form content that I put out, and it's a discussion with a gentleman by the name of Donny, who goes by Standing for Truth on the internet. Now, Donny is a young Earth creationist, which means that he believes that the Earth was created pretty much in its current form about 6,000 years ago. So what you're going to be watching here is essentially a debate, but it's not going to be a debate like you're used to, because I am, in fact, a lawyer. And so my style is very different from what you're used to seeing, in that I'm going to be asking a lot of questions, and I'm going to be demanding answers to those questions, and those questions are going to have a particular strategy to them. And you'll see how it results in Donnie running from the questions and ultimately being cornered and refusing to answer. So that's kind of what you're getting yourselves into. Now, the reason that that I'm doing this, and in fact the goal of this channel in general, is to help give people some more tools to make their points, and in particular to counter ideas that don't have a lot of merit, but where people can be very difficult. In particular, what we're going to be talking about here is, of course, pseudoscience. So young Earth creationism is a great example of that. And the beauty of it is that, whilst it is the case that, of course, we know that they don't have a legitimate point, these young Earth creationists are very cagey, and they're very good at trying to avoid the point. So the reason that I'm fun to watch is because I'm kind of the perfect poison to that particular approach, because I know how to chase them down and force them to answer the questions. And so it provides a very good foil for me to demonstrate some of the techniques that we use in law to make sure that we actually get to the truth. And that's what you're going to be seeing today. So just to give you the final bit of background, Donny believes that, as I said, the Earth was created only 6,000 years ago, and he believes that Adam and Eve are real people that actually existed, and that we can prove that they existed a mere 6,000 years ago by looking at the mitochondrial DNA within the genome. And he thinks that if we use something called a pedigree mutation rate, which is just the mutations between a mother and a child, and we use that to count back, that's appropriate, and that's going to get us to a mitochondrial Eve, as it were, within that 6,000 year time frame. And so this debate is about how that's not the case. So what we're going to start off with here is some preliminary discussion, which I've sped up so that we can get to the good stuff, where we'll slow down and you can enjoy it. So as I said, and I'll pop back in now and then to give a little bit of commentary, but enjoy the debate.
I'm going to assume that we agree on the reality of reality. Then, if there's something that that fusses you about that, then I guess we can go over it. The other thing I want to make sure we agree on is that we both agree that natural selection is a thing, and that it does operate on humans. I'm not asking you to agree that macro evolution or anything, just acknowledge that natural selection exists, that it is a force, even if a small one. You agree with that?
I agree that natural selection being differential reproduction, yes, it's acting. It's not. It's limited for sure. You know, it's mostly good at removing the worst mutants from the equation and amplifying your rare beneficial mutation, but it's almost non-existent when it comes to most mutations, nearly neutral mutations. It doesn't see those. So those are, yeah. So it's definitely there. It's real. But
that's all limited. I wanted. Now, when you're looking at identifying the literal Eve, you're using the molecular clock, right?
Now, one of the things that really helps when you are going to pin anybody down on something is a good command of the subject matter. Now, it's not always necessary. In fact, it's not always possible. And sometimes you are going to have to get the information from the person that you're questioning. but you can be more efficient, and you can more easily detect inconsistencies if you have done your homework and you know what you're going to be talking about going in. One of the ways that Donny wins a lot of these arguments on this issue is that he has a pretty good command of some of the science. Some of it he misunderstands by accident, and some of it, as we will see, he does understand, but and he knows better, but he makes the argument anyway. And I'll point that out to you when the time comes. But just to refresh the memories of everybody who neither just walked out of a high school biology class nor has been nerding out on the internet on this stuff, inside of your cells is something called mitochondria. Now, if you remember one thing from biology in school, it's probably that the mitochondria is the powerhouse of the cell. Now, what you might not remember is that mitochondria actually have their own separate DNA. We think that's because our ancestors swallowed mitochondria back when they were still single-celled, but that they didn't eat them. And then the mitochondria and these early cells then became friends, and they've lived inside of our cells ever since. The technical term for this is endosymbiosis. Anyway, and importantly, because this DNA is not in the nucleus, it is inherited only through the mother. Now, it still mutates slowly over generations, but because there is no scrambling and recombination due to sexual reproduction, it's easier to track these changes over time. And this gives rise to the idea of the molecular clock, quote unquote molecular clock, that which is that assuming that the rate that those mutations occur in the mitochondrial DNA is consistent or consistent-ish, that you can look at two groups and then note how many differences they have, and then just like multiply that by the rate of the mutations, and then determine how long it's been since they split off from each other. Now, the Eve Consensus Project took that one step further and said, why don't we just do that with everyone and figure out how long it's been since any group has split from any other group ever? And that's possible. And this starts to get into something called coalescence theory, which I'm not going to explain here. And even Donny doesn't understand, but suffice to say, this does not mean that there was ever a population of like one or two creatures that couldn't interbreed with anybody else. The so-called mitochondrial Eve would have been but one member of a population. Anyway, okay, so hopefully that's enough background on the molecular clock, and let's get back into it.
Yeah, we're looking at mitochondrial DNA diversity in combination with the observed mutation, right?
Okay. And the molecular clock is based on the accumulation of changes within the genome as a whole over successive generations, right?
Well, yeah, if we're looking at the pedigree rate, right, you can look at
We're
grandmother,
not necessarily
mother,
looking at the pedigree rate. We're looking at the molecular clock in general. The molecular clock is the accumulation of changes within the genome as a whole over successive generations, right?
You can take grandmothers, mothers, daughters.
It's not
You
the
can
question I
count up
asked
the
you, Donny.
differences. Well, I'm going to answer the question however I want to. You can take the differences, you can count them up, and what you typically come up with is a fast mutation rate. But we're not, as creationists, we're not necessarily concerned. And this is where I should ask you: have you read the Eve Consensus sequence paper yet by Dr. Rob Carter in the conventional?
So this is one of the things that Donny's actually good at, is that he starts talking technical mumbo jumbo, and a lot of people's heads just start spinning. And in fact, I cut a whole big chunk of that whole conversation out because it was off topic, and he was trying to divert the issue and stuff like that. So you guys don't need to hear about that. But what you do need to understand here is that there's going to be a debate with respect to the rate at which mutations accumulate. Now, there's two big contenders, two big ideas in the ring here. And one is something called the pedigree mutation rate. The pedigree mutation rate is basically just you take like a mother and a child, or maybe even like a grandmother, mother, child, so either like 2 or 3 generations, and you just look at their mitochondria and you count the differences, and you say, okay, well, that's how much it changes in 1 or 2 generations, and then you just multiply that out. And for a number of reasons that we're going to be going into, that's wrong, because the rate starts fast and then it tends to slow down to something called the substitution rate, which is kind of the longer term, more average rate. And when you're trying to go back as far as you can, you want to use the rate once it's sort of settled down. And that's what we're going to end up getting into and talking about here, just so everybody is kind of on the same page and doesn't get intimidated by the technical discussion, which is being thrown in here kind of on purpose to obfuscate, but that's what's going on.
Mitochondrial DNA. For example, I'm not necessarily concerned with species-wide change, how quickly or how slowly the entire species changes as a whole. I'm more concerned with divergence from the Eve consensus sequence that was published by Dr. Rob Carter in the conventional literature, which is what I'm trying to find here. And on average, it's about 21 mutations different.
Right. I remember that's the average. Yeah. And it's about 140 is a maximum. You have to use the maximum, right? We had that conversation before.
And that should make sense, right? Because what we're trying to do is we're trying to find out like when every group split from every other group. And so we need to know, what the most different group is from every other group, because we want to include them too. We're not just trying to find out what the differences are between the most closely related groups on the planet. We're trying to figure out what the total distance is between the most distantly related groups on the planet. So you have to use the maximum. You can't use the average. I think that probably should make sense. And he knows that too.
It doesn't matter what the average is, because what we care about is what is the most different that we have been, because that's the amount that we have to accumulate over that period of time. Right.
So the question is, how quickly or how slowly do humans diverge from that Eve consensus sequence, in the sense of how long does
and
it
so
take
when you're doing that, when you're looking at how quickly or how slowly those two diverge, you're looking at the changes within the genome as a whole over successive generations, right?
Just keep making your case. I want to find the paper. I got a lot of slides.
No, I want to nail this one down.
Okay, and so then after all of that, then we finally get back into it, and here's that part.
Let me put it another way. Let's see if you'll agree with this, because I think you're hesitant to agree with that statement. Would you agree with this? Would you agree that by looking at how far apart, you take two groups, you look at how far apart each group has drifted, and based on that, we can determine how long it has been since their genes were mixed, which is to say, how long has it been since they had an ancestor in common for that gene? And in this case, that gene that we're looking at is the mitochondrial genes, right?
Not necessarily, because how long does it take to accumulate 140 differences from the Eve consensus sequence? And so we know the mutation rate is fast. And so we know that
Donny, I'm just going to ask you to stop flipping through your slides for a second and listen to what I'm saying, because what I'm saying is the same thing as what I think you're trying to say, but you're not listening to me because you're trying to find something else. Just listen to what I'm saying.
Yeah, go ahead.
Okay, there's two groups. You're still flipping. I can see you doing it. There's two groups. In order to determine how far apart they've drifted using the molecular clock, what you're going to do is you're going to look at how different their respective genomes are, the two groups' genomes. And that will allow you to determine how long it's been since their genes had been mixed together. And what that in turn means is how long it has been since they had an ancestor in common. For example, the mitochondrial DNA. That's true, isn't it?
I think that's a mathematical blunder.
It's difficult because if you look at this
It's-
mitochondrial, if you look at this
That's what mitochondrial
Anderson,
is.
I've given you so
Sorry,
much uninterrupted time to speak.
Sorry.
You've literally spoken more than me. Relax, okay?
Go ahead.
So if you look at the mitochondrial DNA phylogenetic tree,
Some people might wonder why I let him yell at me at that point. I mean, he did lose his temper, and you know what? Really, it's not a big deal, and getting mad about it is just going to derail the conversation. So I'm just going to let stuff like that slide.
that's I think there's some built-in assumptions, because let's take the HVR haplogroup, right? These are a reflection of DNA differences in people groups over time. These branches all go back to a common ancestor, but the branches are different sizes. So we have differing size branches, which means the mutation rate is variable. It's not.
And now watching this, I'm reminded of why it is I interrupted him, because he's off topic again. So we're probably going to speed up at this point.
Not constant, because this tells us that people are picking up more mutations in the same amount of time than their cousins. And so it's not that easy to take two different people groups, count up the differences, and try and figure out when those people groups split from a common ancestor. Let's say if we were to look at, a certain island, or when a population founded a certain part of the globe. Go ahead.
So I put it to you that when we are using the molecular clock, the presumption that we make is that rate at which those differences accumulate is constant, and that's how we count back in time.
No,
and that rate.
I don't agree that it's constant, and that's where I would differ from Dr. Jeanson. Is I believe the mutation rate is not constant, and you can see that reflected anyways, because you have people in the same amount of time picking up more mutations than their cousin, and we know.
So what Donny's referring to here is one of the things that's important to him, which is that he thinks Africans evolved at a faster rate, and he thinks that Denisovans and Neanderthals also evolved at a faster rate, and he needs that to be true for the Neanderthals and the Denisovans to explain how they evolved from Adam, considering how different they are from us, and he needs it for the Africans to explain why it is that they're more diverse than the rest of us. And the real reason is because they've been around for longer, because we all diverged from Africans, but he doesn't want to say that either. At any rate, he is now going to go into silly mode again and start talking about irrelevant things, so we're going to speed up once again.
The mitochondrial DNA, we know on average it's between 20 and 25 DNA differences separating any two people from each other, or the Eve consensus sequence max is about 140. So how long does it take? Rather than looking to species wide change or the substitution rate, how quickly does a species change? We're more concerned with how long does it take to accumulate the max amount of DNA differences. 140 in light of a per generation mutation rate of about 0.1 to 0.5 in the mitochondrial DNA.
Well, but you can't say that, Johnny, because you just told me the mutation rate's highly variable. So which is it? Is it highly variable, or is it relatively consistent? Because if it's relatively consistent, then you can count back and you can get your 6,000 years. If it's highly variable, then you know what? Maybe Eve was a bajillion years ago, and the mutation rate was really slow for a period of time. But I don't think that's the argument you want to make.
It sounds like it's consistently fast, but
so it's consistent.
Some lineages will mutate even faster, in the sense that they'll accumulate more differences based on.
I'm not trying to bust you on. I know that you want to say that Africans are faster, and I know that you want to say that Neanderthals are faster. You know what? I don't agree with you, but we're not even going to talk about that right now. Let's just talk about, except for those, okay? And if you got other examples, say except for those two. Except for those, the mutation rate is consistent, right?
It's still difficult because, again, this is the HVR haplogroup, most common in Europe. And you can see that even amongst them, which are not Africans, it's not Neanderthals, not Denisovans, the mutation rate's variable. And so it
Yeah,
essentially
or
it breaks
maybe those,
the
or
evolution.
maybe those are closer in
Anderson, the
time.
beginnings of your sentences are starting in the middle of mine. It
Sorry,
means
go
that
ahead.
you can't make those evolutionary assumptions based on the evolutionary molecular clock. All we can do is look at the Eve consensus sequence. We know how many mutations removed we are from Eve. Okay. Does
Right.
it take 200,000 years or 100,000 years to accumulate those DNA differences, or does it
Mhm.
just take a couple hundred generations? And I would say the most parsimonious explanation is that we're looking at only a couple hundred generations worth of mutations on this phylogenetic tree. Go ahead.
Right. But see, so what it seems to me that you're saying is not only do you think that the pedigree rate is the rate to use? See, what the conversation I wanted to have with you is whether or not we should be using the substitution rate or the pedigree rate. But it seems to me that you're saying not only do we need to use the pedigree rate, in fact, that rate is useless, and we can't tell a darn thing about when. Like the molecular clock is not a thing. That's what you're arguing with me right now. Do you think that the molecular clock is a thing? Answer me that.
I don't think it's that there's a strict molecular clock.
Do you think there's a molecular clock at all?
Well,
Is it a useful concept?
I think we can point to a rate that is consistently fast and yet variable. And I think that consistently fast rate, in
It
light of
can't
the
be
fact that
variable and consistent. It has to pick. You got to pick one.
So here you can see how Donny's been trying to kind of avoid the thorny issue of which rate to use by saying both that the rate is highly changeable and that it is consistent in that it is consistently fast, and therefore your slow consistent rate can't be the thing. But of course that doesn't make sense. I mean, either it's variable or it's not variable, which is to say it's consistent. So this is kind of one of the first turning points of this conversation, where he's kind of getting pinned down into having to admit either that he doesn't believe in the molecular clock at all, or he's going to have to commit to a rate, and he's going to have to agree that whatever that rate is, there is an average rate which is somewhat consistent, and you'll see that happening here.
So,
Well,
do you believe in the molecular clock? Do you believe in its ability to? Do you believe in its ability to allow us to estimate how long or how many generations removed one population is from another, or do you think that it's got no use at all?
no, I think there's a use. We'd have to look at case by case basis, though.
A case by case basis. So there's no consistent use for the molecular clock. What you're telling me is you don't believe in the molecular clock.
I don't believe in the evolutionary molecular clock that has to
It
assume
doesn't sound like you
very
believe in any
strict.
molecular clock. Do you believe in any molecular clock?
Well, I believe we can make conclusions based
What
on
conclusions can
the
we make?
accumulation rate. Okay.
But how can we make
But how
conclusions
far
based on the
back can we?
accumulation rate if we don't have an accumulation rate that you think is consistent? See, what the problem is, Donny, you're kind of cornered here, and that you're going to have to either say I don't believe in the molecular clock, or you're going to have to admit that you believe that there is a consistent rate. You can say it has a little bit of variation here and there, but
Right.
if we're going to say that there is a rate at which humans accumulate mutations on a per generation basis, that rate is going to have to be something that you're going to have to say is consistent. Otherwise, the molecular clock is utterly useless, and you just don't believe in the molecular clock, which is fine. Which way do you want to go?
I believe it's consistent enough to say that there's only a couple hundred generations on any given mitochondrial DNA phylogenetic tree. Now, when we look to certain groups, we
Okay.
understand that there are factors.
I
at
know about
play.
the Africans and the Denisovans and I mean, I don't agree with you on any of those. I
Well,
think
no,
you're,
again,
but I
even
don't
here,
want to talk about that. I just
even
want to
here,
talk about the
but
regular rate.
I, for sake of argument, I will say, even though, like, let's just say the HVR haplogroup, even though the rate is variable, it's not variable enough to where we can't look at a per generation rate
Okay.
and understand that that per generation rate or accumulation rate takes us back to, let's say in this case, an Eve about 6,000 years ago.
Right.
Less than
So we
10,000
can
for sure.
calculate Eve. You believe that we can calculate how long ago Eve was because we have a molecular clock, and that molecular clock accumulates variations at a consistent enough consistent enough rate to be useful, right?
To be useful enough to say, hey, the on average DNA differences being 20 to 25 is
To be
easily
useful enough to
explainable.
calculate the age or the
I'm
difference in
I'm
number
not
of
concerned with
generations.
species wide. I'm not concerned with substitution. I'm not concerned with how quickly
It,
the entire
if we
human.
take two
I'm not done
groups,
talking. I'm not
go
concerned
ahead.
with how quickly the entire human species changes as a whole. I am concerned with how long. Okay, we can reason together. How long would it take to accumulate roughly 25 DNA differences?
It's probably a good point in time for us to point out that, so to some extent, using the term substitution rate or fixation rate is very confusing. And that's because really what we're talking about here isn't technically the substitution rate in terms of species as a whole change. What we're talking about is the neutral mutation rate. And the thing is that there is this strange equivalency between the neutral mutation rate and the substitution rate. And so we use the same term sometimes for both of them. So just keep in your head that when we're talking about the substitution rate, although Donny does try and conflate this with the rate of species as a whole change, what we're really talking about here is the quote-unquote phylogenetic rate, sometimes called the fixation rate, sometimes called the substitution rate, but it's just this slower rate that occurs after the speed settles down. It doesn't necessarily have anything to do with the spread of that mutation through the population. It just so happens that that speed has a relationship with that rate of mutation, and that's why it's confusing.
From the Eve consensus sequence that you would argue is a coalesce sequence, I would argue it's Eve's real created sequence.
That's the same thing. Eve's real created sequence would also be a coalescent sequence, right?
Right, but you would say that at one point each DNA position there, each allele was a wild type allele that drifted to fixation.
What I
but
would
I
say
am
is
saying that there was no
that
previous ancestors to that sequence, the Eve consensus sequence. It's the created sequence. There's no prior ancestors to
I know. That doesn't matter to me.
Okay.
So, not for this conversation, it doesn't. So, if a change, so we're talking about how far, when we're looking at let's say, for example, an island population, do you agree with me that you can figure out how long a population has been isolated from another population using the using the molecular clock? So, for example, on this tree that you've got, this phylogenetic tree, we can say, look, this molecular clock is going to show that the European group split off from the Asian group about X number of generations ago. And all of these variations that form part of the European clade, those separated from all of these variations that form part of the Asian clade, and that happened X number of generations ago. And the biological clock can tell us that. Do you agree with that?
No, not necessarily, because I do agree that a phylogenetic tree like this is a reflection of the last 4,500 years of human history.
Now, this for me is one of the frustrating things about doing this as a debate rather than a formal cross-examination like I'm used to, is that, you're not in complete control. So you have to let the other person have a turn and go off on these little diversions. And again, that's what's happening here. So we're going to speed up once again.
So let's get back on track then.
Are we talking Y chromosome or mitochondrial DNA?
We're talking mitochondria.
Okay.
Talking mitochondria, always have been. So I want to talk about, so you'll agree with me then that when we look at that graph, you can tell by the branch length, the distance between the different nodes, you can tell how far it has been since, or how many differences have accumulated between the various nodes, right?
No, I think it's too variable. He hasn't tested it. He made the prediction, but he
No,
hasn't
dude,
tested it. He said
that's how the chart is written. You're the one who's quizzing me about this stuff like 2 seconds ago. That's how this chart's drawn. The length of the line is the number of mutations,
right,
and then the number of splits is the number of variants. That's how it
right,
works. Isn't that true?
right. But I'm saying there's so many
Good. We agree
assumptions
on that.
going into this that
Those are
you're
just
going to have
those
to
are
address the amount of DNA differences that have accumulated since the consensus sequence. So what you're saying, you can look to an
Donnie, what you're
island.
trying to
You
do
can look.
right now is just sort of grind this conversation to a halt so that you don't get cornered here. But I mean, we can go around and around and around on this until kingdom come
Well,
if you
dude,
want. I don't mind, but you're going to get
even for
tired.
sake of, I'm not cornered because even for sake of
Not
for
yet.
sake
You're
of
not.
argument,
No.
even for sake of argument, if I were to say you got a strict molecular clock, here's the
Sure.
mutation rate, it's 0.1 or 0.5 per generation.
All
Okay.
right.
If you want to take that back linearly, how many generations does it take to
Let's
accumulate?
assume those
Let me
things for the
finish.
sake of
So if
argument.
you,
Thank
for
you.
sake of argument, if you want to say that it's just this linear clock, okay,
Sure.
0.5 per generation in the mitochondrial DNA. How long does it take to accumulate 140? Just a couple hundred generations. There you go.
Sure.
That's the
So
creation
yeah,
time.
just take that position. Thank you. That's all I expected of you.
Dear God, why is my life so hard?
Whoa, hold on, what? It's hard?
Yeah, it's really hard.
So if a change doesn't accumulate in a successive generation, then it's not going to matter in terms of the molecular clock, though, right? Because it's not going to show up in the genomes of those people however many generations down the line.
Are you
right?
saying in a single generation mutation?
I'm saying that let's say, I'll give you an example, right? If I get cancer of the pinky toe 10 years after my kids are born, that mutation won't accumulate in the subsequent generations, right?
If you get
It's not going to matter in terms of the pedigree rate. It's not going to matter in terms of it's not going to matter in terms of the substitution rate, because that mutation in my pinky toe, it's going to stay in my pinky toe. It's not going to make its way into my kids because first of all, it's in my pinky toe,
right?
not my sperm. I know it's the basics, but just agree with me if you agree with the basics.
Yeah, you
That's
got
not going to
somatic
get into the
cell
next
line
generation,
mutations,
right?
like your skin cells, they're not passed on, but
Yeah.
your reproductive cells, your germ cell lines, those are the mutations that are passed on, right?
Right. It's only the germline cells that are gonna matter, right?
Right.
Okay. And so we have to realize then that not all of everyone's genes are going to make it down the line into the next generation too, right?
Right. Well, only the ones that are passed on through the germ cell line, right?
Well, so there's the ones that are in the germ cell line, but then also so I have 2 kids, right? Not all of my genes are in those 2 kids, right? There's going to be a random mix of half my genes that are in one and a random mix of half my genes that are in another. And if we were to look at both of the genes that I passed on to my children and add them together, it's not going to add up to being all of my genome, right?
Right.
Okay. So luck and natural selection are also going to play a role, right?
Natural selection isn't going to play a role on the nearly neutral mutations. They're going to drift.
No, only on the ones that are selectable, but it will play effect.
The one's big enough,
Yeah. All right. Now, if we I want to look at how Jensen calculated or looked when he did his pedigree mutation rate. He used a rate from a paper called Ding et al., right?
Right.
And that was a rate that was calculated between parent and offspring. So from mitochondrial DNA, which is inherited only from the mother, that was the number of differences between the mother and her child due to mutation. He used a study from 2015 that compared mitochondrial DNA from between parent and offspring that was extracted from their blood samples, right?
Right.
Okay. And blood, of course, is a somatic cell. it's a working part of the body rather than a germline cell, right?
Right.
Okay. So it's a cell that it's not a cell that would be used to transmit genetic information to subsequent generations, right?
If it's a somatic cell,
It's a blood
mutation, no.
cell.
Right.
Okay. And somatic cells divide throughout the life of a person in order to maintain homeostasis, right?
Yes.
Okay. And as those cells divide, they're going to start to accumulate mutations of their own, aren't they?
Of course.
Okay. But that's not true of a woman's eggs, which she's actually born with, right?
Yeah.
Okay. So if you measure the differences between somatic cells in an adult and their offspring, a bunch of those changes are going to be changes associated with mutations that occurred during that person's lifetime that are not going to be passed on to the next generation, right?
Right.
Okay. In fact, they'll be from three sources. You're going to find mutations from three sources. One, germline mutations. Two, somatic mutations that occurred in the parent. Three, somatic mutations that occurred in the child, right?
Right.
And those differences from numbers 2 and 3 could in fact be significantly more than the amount of change that you're going to observe in the germline, right?
It could be a higher mutation. So the mutation rate that was derived from the Ding paper could be
Yeah.
higher than it really is because of these factors.
Right. In fact, it could be significantly higher, right?
Of course, if it's counting somatic cell line mutations, then yeah.
Yeah. And the Ding 2015 study that Jensen used when he was calculating his mitochondrial Eve pedigree mutation rate included all three of those things, didn't it?
Right. I debated Dr. Dan Stern Cardinal on the same topic 2.5 years ago.
Yeah.
You're not telling me anything I don't know.
Yeah, I remember before I told you guys that there were some things that he just misunderstands, and there are some things that he does understand, but he makes the argument anyways. Yeah, this will be one of those things.
All right. Well, I mean, here's the problem I have, though. If you were to factor out those other changes, the somatic changes, then you're not going to get a 6,000 year timeline, are you?
Well, if you're just basing it on that one study, have you looked at, the Parsons study?
Pick a study that does that. Pick a study that doesn't include the somatic mutations, and that number goes above 6,000 by a lot, right?
Are you familiar with the Parsons paper?
Nope. I am familiar with lots of other papers, and none of them say that 6,000 is the one. The only way that Ding gets you to 6,000 is because it includes a significant number of somatic mutations, right?
Here's the Parsons one. I don't look to the Ding one. So here's the Parsons paper.
So you don't use that pedigree mutation rate.
Right. Yeah,
Mhm.
I agree.
Now, that little mumbled concession may not have seemed like much, but that was actually quite significant because the graph that we've been staring at through most of this debate is taken from a book by a guy named Nathaniel Jensen, and it is based on that study. And that's the ideas that Donny has been defending for years now at this point. And it is the only study that's going to get you to a 6,000 year rate. And as we shall see, neither the Parsons study nor any of the other studies are even going to get you close, which is why Jensen used that study, why he misrepresented that study. And Donny's just admitted that you can't use that study. So that's actually quite significant.
So
Okay.
the Parsons paper, TJ Parsons.
Yeah.
So this is Ann Gibbons. This is a commentary on the Parsons paper.
Mm-hmm.
And the Parsons paper talks about how the mutation rate, a lot faster than the phylogenetic rate, pointed to mitochondrial Eve 6,000 years ago.
Having now read that Parsons study, I can tell you that no, it does not. the way this works is that Donny is saying that there are the 3 women who were the 3 daughters-in-law of Noah, and that they represent all of the mitochondrial DNA, and that they lived about 4,000 years ago. So that would be about 200 generations ago, if you assume like a 20-year generation time, which is, I think, pretty tight for women to be having kids, more than one kid by the age of 20. So in order to get that rate, you're going to need in order to get the number of changes that you see by then, you're going to need a mutation rate of 0.7, which is to say, in order to get 140 mutations within 200 generations, you're going to be needing to have almost 1 mutation per generation, right? 0.7 mutations per generation. And the Parsons paper has a rate of 0.028. So that's more than 10 times slower than you need, even for his super fast Parsons mutation rate. So Parsons isn't going to get you there, and it's like the fastest one that you're going to be able to find. All the other ones are even slower than that.
And the data was so good, the methodology was so good, that this contributed to the development of mtDNA research in the FBI. So the FBI has actually adopted this rate or methodology in getting the rate, and it was fast, very fast.
Yeah,
Went
well, the
back
FBI
into-
is going to use is not going to be using the phylogenetic rate either now, is it?
right.
Right.
They're
It's going to use a pedigree mutation
rate.
rate, because they're caring about the fastest possible mutation rates, because they're going to be discounting things like selection, right?
Right, exactly.
And the longer the span between parent and child. If you do 3 generations, it's slower. You do 6 generations, it's slower yet. You do 9 generations, the mutation rate is slower yet. Isn't that true?
Well, the Parsons paper used the pedigree rate and showed that it was very fast.
Yeah,
and took
for a very
Eve
short
just
period.
6,000
I'm not
years
familiar
ago.
with the Parsons paper, but I bet you a nickel the Parsons paper uses a rate like 1 generation, 2 generations maybe. And if you were to do 3 or 4 or 5 or 6 or 9, then the rate slows down, and it slows down quite quickly. Isn't
But
that true?
It would be right, but I think it
Have
was
you
a
looked
trio.
at any other papers
Pretty sure
besides the Parsons
he
paper?
looked at a strong. He looked at a good amount of data. It wasn't just a 1 or 2. I think it was a trio. So grandmother, mother, daughter.
Mm-hmm.
So I think it was
So 2
three
generations then, or 2
But from my
steps.
understanding,
And if you do 6 or you do 9 or you do 12, it gets bigger and bigger and bigger, doesn't it?
well, according to if evolution's true, your
not according to if evolution is true, just based on looking at these pedigree mutation rates. If you look
Right.
at various pedigree mutation rates, the longer they go, the more generations they do, the longer or the slower the mutation rate, right? It slows down and down and down, doesn't it?
Well, but again,
So yes or no, Donny?
well, no, because the
It doesn't
neutral
slow down?
mutation rate, the neutral mutation rate that you would get, let's say you
This
take
isn't about the neutral mutation rate, because we're not discounting selection. Selection is probably one of the biggest factors here after we've accounted for the somatic mutation rate, which was your first big problem.
It's the mutations that are
And
subject
selection
to
is your
drift.
second problem, because as selection comes in and as drift comes in, it eliminates more and more of those pedigree mutations, and fewer of them get into the subsequent generations as we go more generations
Right.
down the road, and it slows right down. Isn't that true?
I wrote a technical article with Chris Roop on this. Have you read it?
I've read a number of your articles. I think I have read that one, and I think that what I'm saying is still the case, and I'd like you to agree with me. You keep on changing the subject. Isn't it true that the more pedigrees, or the more generations that you look at with a pedigree mutation rate, the slower that mutation rate?
No, because most
No.
mutations are lost, but many are not. And so in the evolutionary time scale, your neutral mutation rate, those mutations that are not subject to selection, it is going to equal or be close to equaling a fixation rate.
That's right.
This is called
It's gonna,
fixation
it's
equilibrium.
that. And what's another name for a fixation rate? It's called the substitution rate.
Exactly.
So
And so
as
the substitution
selection
rate
has
is
its
therefore
effect,
by definition
and
high.
as it slows
It is
down, it
fast.
approaches the substitution rate. But I'm not even forcing you to go there, Donny. All I want you to do is admit that if you look at just pedigree mutation rates, if you do longer generations, it slows down. And I put it to you that it slows down to a point where it eventually reaches the substitution rate, because now you're dealing with nearly neutral mutations finally, because selection has factored itself out.
But here's the
Right?
thing. According to your
Right,
model,
Donny, though? But
listen,
right?
no, I'm going to explain some basic things to you. According to your
Go for
model,
it. You mean the pedigree mutation rate for how many generations?
the observed mutation rate.
For how many? The observed pedigree mutation rate for how many generations?
Well, you tell me. When Fatuma says that the neutral mutation rate
Okay, so
equals
if you
the
want to take the
fixation rate,
neutral mutation rate, I'm fine with that. The problem is when you take the
how do
pedigree mutation
you
rate for
derive the
one
neutral mutation
generation or for
rate?
3 generations.
Let's
You
say
take
3
it for
generations.
14 generations or 20 generations, and you know what? It's going to approach the fixation rate, and then we're good.
So once again here, Donny is trying to obfuscate. He's trying to confuse the issue. And I think in this part, in fairness to him, there's a degree of genuine confusion here. And this alludes, again, back to that point that I made before about how it is confusing that we use the words substitution rate and fixation rate and phylogenetic rate and neutral mutation rate here in some sense semi-synonymously, although they are different concepts. And that's because in this context, they all equal the same thing. And now Donny is trying to conflate those various issues. So what's going on here is that we're getting into a technical discussion because Donny wants to try and confuse the issues. And, I'm just kind of insisting on, look, I understand those confusing issues, but they don't actually matter. What matters here is if you look at the pedigree rates, they slow down. And that's the thing that you have to wrestle with. And you can see what I do is I keep bringing him back to that simple point, because that's what matters. All these other interesting things that he's getting getting wrong in an interesting way, they don't matter to the central issue. So just keep that central thing focused on your mind.
Pedigree mutation rate is going to be is going to approach the substitution rate as we get to 20, 30, 40 generations.
But
and you can observe that.
That substitution rate would be fast because you have, according to the evolutionary model, a long time scale with faster rates of fixation because you have a smaller population. And so the last
what that substitution rate's speed is going to put, where it's going to put mitochondrial Eve at that fast rate? About 1 to 200,000 years back.
No, that's based on the phylogenetic method. That's based on
the phylogenetic method is the fixation rate. That's the same. These are the same words. They mean the same thing.
I understand that they're used interchangeably and synonymous, but you're concerned with species-wide change. I'm concerned with how long does it take to accumulate these DNA differences from the Eve consensus sequence. And here's the thing.
We're both concerned with the same thing.
You admit it. Smaller populations are going to have faster rates of fixation. After the flood, you have humans existing as how many people after the flood?
Your effective population size is 5.
Right. So how fast?
5.
How fast is fixation going to be after the flood? With 5 people that are reproducing. Fast or slow?
On the record, that was like 45 minutes.
Okay, I'm not. That was basically a debate. I'm not going to shut it down at 4 hours, but what I'm going to do is I'm going to eat some food. T-Rock, act as because I know T-Rock and Grayson want to have some impromptu debate, so I'm going to keep it going. But but let's keep it cordial. Let's keep it respectful. Passionate's fine. Me and my boy and Mr. Anderson, we duke it out. We give the audience a good show.
Donny, I got respect for you. I'm not going to try and give you the gears. But before you go,
yeah, I'll give you the last word. Go
one
ahead.
question, and it's a question I've asked you a number of times before. You will agree with me that as the pedigree rates increase in number of generations, the rate at which those differences, the rate at which those mutations accumulate in the end ancestor decreases significantly and approaches the fixation rate, right?
I will give you an answer to that on another, because I have to study it a little more, because I don't know how that applies.
Boom! Lawyered.
Everything we've been talking about in terms of your neutral mutation. So when Fatuma says neutral mutation
Okay.
rate,
You know what?
And there you go. That's pretty much the end of it. He says a couple more things, but they're not really relevant to the discussion. So you can see in the end what happened was I had to chase him. I had to chase him for a long time. And I had to pin him down first with respect to the fact that there is such a thing as an average rate that can be used to calibrate a molecular clock. Finally, he admitted that. Then we went into how the study that he's been using for years doesn't work. Then he pivoted to this new study, which now, having looked at it, we know also doesn't work. And then ultimately, the thing that he couldn't wrestle with was that even if you look at those studies, if you look at studies that have longer and longer generation times, more generations, that rate slows down, and it slows down until it approaches what they call the neutral mutation rate, sometimes called the substitution or fixation rate, which is the rate that they use for phylogenetics, and the rate that puts mitochondrial Eve way older than 6,000 years. And so hopefully you can see, kind of having gone through this whole process, how the argument was constructed and how on a step-by-step process we were able to force somebody who was very resistant to face a truth that he kind of knew all along. But yeah, that's how we were able to sort of get to the truth. So if you like this content, then please consider subscribing and leave a like on the video. And if you like the content, then say something nice. And if you can't think of anything nice to say, then say something mean. Tell me what you think of my shirt or something. Yeah, that's it for me. So until next time, thanks for watching.